And the reason why I took a little bit of time with the background of these two scientists is to give you an idea of where this information and knowledge is coming from. It's not sort of backyard stuff that somebody's made up mumbo jumbo. There's a lot of good hard science behind this work, and this is why you can be comfortable when you start using some of these protocols. Now let's get into the story of the cytochrome 1b1 enzyme. This enzyme is very highly expressed in cancer cells, and they've found this from human biopsies and all types of cancer tissues, breast, prostate, you name it, they've looked at them. And also from immunohistochemistry studies. The enzyme is basically undetectable in most normal cells, but there are some trace levels in some normal cells. Just to give you an idea of what you see through a microscope when you go sticky beaking at a tissue sample, this is in the case of esophageal cancer. They used a purple stain plus a black stain. The purple stain stains normal tissue, the black stain the cancer cells, and you can see here quite distinctly the black stain was specific for that enzyme CYP1B1, and you can see them standing out quite distinctly. So, but the normal cells don't have that any of that stain because they don't contain the enzyme. And this is just a slide to show you that they found this particular enzyme in all the various cancers: bladder, brain, breast, colon, connective tissue, and so on and so forth. And here is another example of some tissue slides. On the left-hand side, we have prostate cancer tissue sample. On the right-hand side, we have normal prostate. And because of the two different stains, we have the blue stain staining the normal cells, and we have the brown stain staining the cells that contain that enzyme, the 1B1. Yep, so it's quite clear. Cancer cells have it, healthy cells don't. And again, here's just another slide showing where they've studied all these various types of cancers and have found the enzyme expressed in all of these cancerous cells, as well as in metastases and precancerous cells. So it's pretty widespread through these diseased cells. This is a nice little picture of what's happening visually within the cancer cell and normal cell. On the left-hand side, we're looking at the cancer cell. Up the top, you've got the CYP1B1 gene, and the information is being read out from the gene, and the microRNA leaves the nucleus. Now, the microRNA is like a blueprint, which then travels to the ribosome, and that blueprint is then used to build the CYP1B1 protein. But if you look at the normal cell on the right-hand side towards the bottom, we have the gene, we produce the microRNA, but there's a feedback mechanism, there's a DNA interaction, and that ribosome production of the protein is blocked. So therefore, you don't produce any of that particular enzyme within the cell. And now enters the Salvestrols. They are natural phytochemical anti-cancer prodrugs. They've got a unique mode of action. They're activated by the CYP1B1 enzyme in cancer cells. They stimulate automatic cell death, apoptosis. They kill cancer cells without harming normal cells. Pretty important stuff. We find them in fruits, vegetables, and in herbs in our diet. And historically, Salvestrols probably have helped to protect us against cancer for millennia. Unfortunately, now they're really low in our dietary intake. Just looking again briefly at the what we're talking about a prodrug, on the left-hand side, we have the molecule of Salvestrol, which is harmless to the cancer cell initially. And then within the cancer cell, it's acted upon by the CYP1B1 enzyme, and then you create a metabolite. So it's been changed a little bit. What they've done is on the left, from the left-hand side, right-hand side, you've removed a hydrogen and carbon atoms, and you've stuck an extra oxygen on. And this is just goes to show it's only a tiny change, but it's a change enough to trigger the whole process. So as we can see again here, that on the left-hand side, the Salvestrols can quite happily enter and leave a normal cell. There's no reaction because there's no 1B1 enzyme. But on the right-hand side, that initially inactive and harmless compound becomes an extremely potent cancer-killing metabolite. And what it does, it interferes with the ability of the cell to grow, survive and divide. And so the cell undergoes automatic cell death, apoptosis. So looking at tyrosine kinase and apoptosis, just a quick overview. Kinase is a type of enzyme that transfers phosphate groups to specific proteins, protein substrates. By attaching these phosphate groups to the proteins, this is an important mechanism in communicating signals within the cell and also in regulating cellular activities such as division. Protein kinases can become mutated, stuck in the on position, and cause unregulated growth of cells. And this unregulated growth is a necessary step for the development of cancer. However, cells treated with Salvestrols experience tyrosine kinase inhibition and undergo programmed cell death, apoptosis, and that's exactly what we're after. Because we need to be able to remove damaged or dangerous cells from the body when the body thinks that they could be a problem. And this is what's supposed to happen, but unfortunately with cancer cells, they become immortal. They live forever. They keep multiplying instead of going away like a good little unhealthy cell should. And this is what we're aiming at with the Salvestrol and the cytochrome 1b1 process. Now look at some common sources of the Salvestrols. You find them in apples, blueberries, cranberries, grapes, oranges, strawberries, vegetables such as avocado, Brussels sprouts, capsicums, cabbage, cauliflower, olives, artichoke, basil, dandelion, milk thistle, parsley and thyme. And interestingly enough, that a lot of these compounds are actually towards the outer layer or the skin of the fruit and vegetable. That's why it's so important if you're going to make juices up, for example, to get more Salvestrols, as you often do if you want to be healthy, but also when you're working with cancer patients in their diet. You need to get like a decent sort of juicer which will actually extract these Salvestrols from the entire plant. Here is a nice little chart which shows you the relationship between Salvestrol dosage and the number of cells killed. It's done in tissue culture or human cell culture, and as you can see, the cells that contain the 1b1 enzyme are killed off at a great rate as you pump more and more Salvestrols in. If you look at the healthy normal cells, basically unharmed at any dose. When they've done a similar test again, you'll notice that the light blue dotted line are the cancer cells without the inhibitor. So the enzyme activates the Salvestrols, but when you look at cancer cells with an inhibitor, you'll notice that the cancer cells are not killed, and they're basically as immune as the normal cells. And that's why it's important too when you're working with people battling cancer to avoid anything which is going to interfere with the function of that 1b1 enzyme. This is the view through a microscope of cancer cells that have been treated with Salvestrols. You can see that there are a couple of cancer cells that haven't been affected. There are 3 in fact, and there's one where the cell has succumbed to the Salvestrols, which is outlined in yellow. And here is that same slide. In this case, the light which is lighting up the sample has caused fluorescence to take place. What they did, they stained the DNA of the cancer cells a nice bluey purple, and the cytochrome enzyme was stained red, and you can see the arrows pointing to the enzyme. Now if you look at the which has started to break down, it's what they call broken down or condensed DNA. It's undergone cell death by apoptosis, and the CYP enzyme is about to attack the other DNA in the other cells, and this is what takes place within the cell. So it does this by inhibiting the tyrosine kinase, which we mentioned is involved in cell growth and division and cell regulation. So that once you block that, the cells go into a self-destruct mode. So it's a different mechanism, completely different to an antioxidant effect. Let's look at now why we need protection. 1 in 3 of us will be diagnosed with cancer during our lifetimes, and 1 in 4 will die from cancer. Cancer starts from a single normal cell and then starts to multiply. 1 becomes 2, 2 becomes 4, 4 becomes 16, and so on. And when you're looking on the left hand side here, that is the number of cells. So 10 to the 1 is 10 cells, 10 to the 3 is 1,000, then a million, then a billion, then a trillion. And a trillion cells will make up about 1 litre volume of cancer cells. And when you get to about the litre stage, that is fatal. And it may be a primary tumour coupled together with metastases and secondaries. With current medical techniques, the tumour needs to mass at least 1,000 million cells, or in other words 1 billion cells, and that tumour size is about the size of 1 cubic centimetre, which is, again, if you look at your little finger, about the top half of the top joint area. So as I mentioned, you need at least 1 billion cells for detection, and up until that time there are no symptoms, no lumps, no pains, no coughing of blood, nothing visible to scans. This is what we call the silent phase of cancer, and in fact the silent phase takes up to 50 to 75% of the growing time from the start of the cancer until death. By the time we are diagnosed with cancer, most of the options for dealing with the cancer are no longer available to us. It's too late. Survival after the diagnosis of cancer is a real lottery. The American Cancer Society in 2000 figures released research, and so the 5-year survival rate, that means being alive 5 years after the detection of the cancer, depends upon the type of cancer you have. Your survival rate can be as high as 98% or as low as 68%. And for example, the best cancer to find that you have if you've got to have a cancer is testicular cancer, and that's what Lance Armstrong, the Tour de France winner, on quite a few occasions. Yeah, he had testicular cancer, so he was in that lucky group who had the 98% survival rate. Whereas the worst cancer for survival is pancreatic cancer, and 6% are lucky to survive. And a couple of sufferers of the pancreatic cancer was 2 famous people, Luciano Pavarotti, the famous opera singer, and also the movie star Patrick Swayze. Taking into account all cancers, an average of 50 to 60% will survive. Now, it would be nice if you were told when we had a cancer that you could choose the type of cancer. So we can't do that. What we should really do in that case, look at the green section. This is before it gets to the stage of being detectable and being pretty nasty. We need to work on the cancers before it develops to a stage where we can't get rid of it. The work of Professors Potter and Burke is leading to a much newer and much more effective method of cancer detection called proteomic. And what they've done is they've been able to identify a protein fragment of the cytochrome 1b1 enzyme that is lives only in cancer cells. They've been able to look at this particular fragment within the bloodstream. This is really good too, because it means that you don't have to have surgery, don't have to have biopsies. All you need is a simple blood test to be able to determine whether a cancer is present. Now, with the absolute best methods that we have today, you have to wait until there is a billion cells present before it can be detected, and even the absolute best methods is only slightly below 1 billion. 10 to the 9th. Now, with the proteomic method, where you're looking at these protein fragments of the cytochrome 1b1 enzyme, they're now being able to detect the cancer cells at 1 million, or 10 to the 6th. And when you're looking at time, it gives you a whole lot more time before it gets to the stage where you can do hardly anything with the cancer. And that's what we need, time to work on the cancer to kill it. Okay, we can all see that we need protection, so that a cancer which we cannot even know we have never gets big enough to be diagnosable. So what can we do? Many breast cancers found in routine screening are destined never to harm patients. More old men die with prostate cancer than from it. Cancers that are slow growing and staying small are not always dangerous. We need to cull the cancer herd, slow down the cancer growth, kill some cancer cells, and do it every day. We just need, of course, the more you cull the herd can't keep growing and multiplying. And so what we can do is keep it to a stage where we can keep it under control. And that's where the Salvestrols come in. We need protection beyond antioxidants. Antioxidants are good, they're helpful, they help in developing cancer, but they're not the only answer. On the left-hand side we have the blue arrows attacking us, and we have our antioxidant protection. But every now and again, some of these free radicals and damaging stimulations are going to get past and affect a normal cell, which can then trigger it to become cancerous. And in fact, Professor Bruce Ames, who is the most important cancer scientist living today, says that in our lifetime, each cell of our body is subjected to 150,000 hits to its DNA. This is easy enough to cause cancer without antioxidant protection. But unfortunately, the problem isn't just the situations such as the free radicals and ultraviolet light that can be mopped up by the antioxidants that causes problems, and scientists believe that about 80% of all cancers are due to assaults by many chemicals and some viruses, and antioxidants can't protect against these.